Anyway, I was going to make an announcement, and that is that many of you are getting your vaccines, which is very, very good, and I'm very, very glad that you are. And I'm getting a number of emails from students saying that they're trying to pick a day when they can have their vaccine, when they can take their vaccine, which is not going to make them feel drowsy, to come to this class. Now, while I feel very, I'm trapped, I'm flattered that you're making your arrangements around this course, my advice would be, don't worry about this course, just get your vaccine whenever you can get it, and don't worry about this class in any way, because I think the vaccine is infinitely more important. So what I'm saying is that if there's a lab or if there's a test or something coming up, not that there's any tests anytime soon, but if there's some project where you have to be here and you're deciding maybe I should be in class and get the vaccine some other time, I would say get the vaccine, let me know whenever, like even after the fact, and we will make arrangements so that you can get your work done, and I'm not going to give you a hard time about having it late. So what I'm concerned about though is that you get your vaccine. That's I think that's a very important piece. And so don't worry about this class. In fact, I would wager that don't worry about any class. I would say the vaccine, your vaccine should probably take precedence over everything, because once you have that, then you can start putting your life back together, and then you can start, you don't have to worry about organizing another meeting, and it just makes everything so much more easy. So thank you to those of you. I think over the week, for instance, I had I think maybe 5 emails asking what was in store for class because they were afraid that they were going to get drowsy. And by the way, when you have your vaccine, you do get a little bit drowsy. But if you feel drowsy, then don't worry about coming to class, because you know that all your are online. All the classes that we have here are recorded and they're on YouTube. So don't worry about any of that stuff here. And so for the 5 emails that came through this week about planning for vaccines, get them done whenever you can. The sooner the better. And if you feel drowsy and you can't come to class, so be it. That's fine. Don't worry about coming to class. In fact, it's probably better that you rest and let yourself just relax for a bit. But anyway, so what I'm saying is I'm very, very flexible, and I don't want you to feel that, I want you to be here, don't get me wrong, I want you to be here, but I'm not going to say that you must be here even when you get your vaccine, because I know I was also drowsy when I had my vaccine, and so that made me feel really kind of like woozy, and I felt like, how can I concentrate, how can I give my studies my best. I would say just take the day off. Just relax, let your body get over the vaccine, let yourself relax, and then come back the next day and you'll be much, much more focused and much in a better state of mind to do the work. So there, I've said it. I'm very, very flexible after the fact or whenever it is. Just let me know. And don't worry about this course in terms of your vaccine planning. Just get the vaccine and then it'll be fine. So I hope that for those 5 mails that have come out on Tuesday, Wednesday, and yesterday, I didn't respond because I was going to make a message. I was going to actually say something in class, but that's hopefully your response. Get the vaccine, don't worry about the course. Or don't worry about any course, maybe, just do it. Anyway, if you have any questions, let me know, and we can work around any concerns about class deliverables or anything that might be on your mind. Just let me know. Okay, now, what I would like to say is I'm very excited about today's class, as I usually am. People actually have asked me, why are you always so excited about bioinformatics? I'm excited about bioinformatics because I think that we have a really good class. I think that this is to me very exciting stuff, and I hope that by now you have picked up on the excitement in bioinformatics. But I hope that also you have realized that there are many, many, many types of careers which exist in bioinformatics. Careers that are based in biology, careers based in statistics and math, careers based in writing software, in computer science. There are many, many different ways to get into bioinformatics. And so you might be, in fact, I had a conversation with one of you fairly recently where you were saying that you're coming from a computer science point of view. You're interested in becoming a bioinformatician, but maybe your biology isn't as good as you think it should be. That's okay, because when you're working on projects, then you would learn about that specific biology, but maybe your actual involvement in the project would be to build a database, or to build software, or to build some kind of, to integrate an algorithm, for instance, or to manage the algorithm, or to update software, to build a tool, to enable people to use the tool. You know, there are many, many facets involved here. And so just because you think that perhaps your biology isn't good enough, that's really no reason why you should keep yourself out of this. I think you should invest yourself and see whether there's more opportunities in bioinformatics than you think. And just because you're not, maybe you might count yourself as someone who's not great at math, that's no reason why you shouldn't join an area like this, because you can learn the math if you need to, or maybe you're not really even worrying about the math, maybe you're worrying about something else. Anyway, so there's a bunch of stuff going on in bioinformatics, and I hope that you've kind of picked up on all the fun stuff that's available. I mean, there's room for everybody, which I find especially exciting about being in this area. That there's so much room for so many people, and so many things can be done. I'm going to kind of zoom to where we ended up last time. What we were talking about is basically, this is our summary. We were talking about gene sequencing, and gene sequencing is, I think, actually very impressive as a tool in bioinformatics, or seemingly a tool anywhere. Remember what gene sequencing is? Gene sequencing is to take one strand of DNA, which may be slightly unknown, or maybe we don't know where this has come from, or maybe we want to look at relatedness, or maybe we're not exactly sure whether this is a sequence that we've already seen before, or that we don't know, but whatever it is, we have a genome which we don't know anything about, or we'd like to sequence, and we'd like to learn more, and so we'd like to kind of get the actual, you know, the DNA text, or DNA in text from this genome. So in other words, we have some molecular data, and then we want to turn that into, like, you know, computer science data, like strings. And so how you do that is you isolate the DNA from the organism, you clone it, so you make many copies of it. And then those copies that you have there, those are all long one sequences here. Those sequences are actually too long to actually run through a machine to turn out the A's, the T's, the G's, and C's. It's just too much information, it's overload. And so what you have to do is you have to take those long sequences of DNA and you cut them up into little pieces, chop, and make them into tiny pieces, like just little chunks of the code. And those pieces are called the reads. And those reads are basically like micro jigsaw puzzle pieces. I mean, they're very, very small in terms of the entire jigsaw puzzle, which is the genome. So then you start assembling, you start putting those reads together. You have to analyze each read, and you start joining the reads together in a way that allows you to make bigger and bigger puzzle pieces, and then eventually you can figure out how those puzzle pieces will fit back into the main puzzle, and will allow you to have a completed genome. And the reason why you have to actually put these pieces back together is because it isn't information overload, but as you're putting the pieces back together, you're keeping track of the order that those pieces actually came in. And to figure out what that order was, you have to work with something called overlap. Now here, the overlap can be seen in these graphics, and the overlap here, if you're looking at this one in the middle here, that chunk, let's just say, this chunk and this chunk above it and this chunk below it, that middle piece is what we call the overlap. And so we're using that as a kind of an anchor, if you will, to position these other pieces which were floating around in the experiment, but we found out that where they're supposed to go. We position them there. We say, according to this overlap and all the overlapping pieces before it, we think that this is the spot where they go. And it's only that way that you can kind of figure out, like, what the actual final sequence of DNA was that we're working with. But the thing is, though, that the question that I always asked when I first saw this method is, why do we have to go through all these unnecessary steps? Like, why do we have to do all this stuff? Like, why do we have to, if I can get out of my thing here, but why do we have to cut up the DNA into all these, or clone the DNA into all these copies, and then cut them up and then reassemble everything? Well, it's information overload, and that means that there is so much information, so many A's, T's, G's, and C's, that the technology that we have now is just, it is nowhere near being able to take in a strand of DNA and start kind of like piecing together, you know, like pulling it through like a piece of yarn and piecing together what the code is. And so you have to have a system like this. And even then, I mean, it's like, I don't think that, I mean, think of the logistics actually of grabbing one one molecule of DNA and pulling it through some kind of computer system, how could you? It'd be all knotted and cut up at the end, and so you probably wouldn't be able to grab the end of the DNA and pull it through, in its entirety, and read the bases. But when you're working with these tiny pieces here, yes, there is much more computation involved, but it's also feasible. That means that you can actually do this. You can actually find these little pieces floating around in the soup of your experiment, but floating around, and then you can pick them out one by one by one, and then every time you pick out these small reads, you can figure out what the code is that makes up those reads, and then once you have that, then you can start figuring out how they all combine together algorithmically. I find this really exciting to see that, but it does, on the outside looking in, it does seem like a bunch of wasted steps. Like, why would I need to do this? It's just because there are too many difficult logistics involved with grabbing an entire sequence of DNA that comes right out of the nucleus and figuring out what the base composition is. All right. So anyway, then we talked about, like, for instance, the coverage and the ordering, and this is what I meant by the overlap, which is, very important stuff. But it's almost, I would say it's like it's genius stuff, actually, to use the whole notion, for instance, of, like, for instance, down here, the whole notion that that you can use, you can take all these little pieces and build a computer software package that determines, okay, here's this overlap, which is this TTTT, ACCAA, and find another sequence of DNA, which is TTTTACCAA. That's not another sequence, but that's another read, and figure out that those two actually do connect with each other. They're actually, they're two different copies, but they suggest how the code is written at that point. And for instance, like down at the bottom here, you can see that that's what that code is all about. And so you just have to go through and do that from the very, very start, or, you're basically trying to find, when you're finding these overlaps here, you're trying to find as many overlaps as you can, and then you have to figure out from these clumps, like, where the positions are inside the sequence itself. And so this takes some time, and so I find it actually really exciting to think that there's an algorithm that does that. Of course, now we have algorithms that do everything, but to think that there's like a kind of a logical approach to actually figuring out how to solve a puzzle is actually something quite exciting. When I was younger, I used to spend a lot of time solving jigsaw puzzles with my sisters, who used to beat me every time. Yeah, I'm still thinking about it. Yeah, they did. So they'd work on theirs, which is like 500 pieces. I'd be working on mine. They'd say, "Oh, I got mine done in 3 hours." I'm like, "Oh God." Anyway, but still, but I was always excited to see, like, is there a better way of putting these puzzles together? And then I discovered this. I'm like, oh my gosh, it's amazing. Anyway, so that's the largest overlap, and so you're using overlaps basically to figure out how these pieces come together. And so you're really just looking for the largest chunk of this, of these two reads. But one more thing I will mention, though, going back to this overlap technology here, I'm hoping that you can kind of see that this is yet another application of sequence alignment. Remember how much time we spent on sequence alignment? Two sequences. How similar are they? Is it locally? Are we globally aligning them? Are we locally aligning them? How do we figure out how sequences are related? In this case, this would be a local alignment, because you'd be able to find out that, like, for instance, in these sequences here in the very, very center, this CCCC or CCCGA, that would be a chunk that would show up in a local alignment saying that these two sequences are the same at this position here. Of course, there may be similarity somewhere else, but here it's the largest chunk of overlapping of similar material. So therefore, we'd say that would be the overlap, and that's another sequence alignment algorithm that's at play. And so again, it's like alignment. It's like everywhere. And so that's kind of how this is. And so you're using alignment here to figure out the largest overlap or the longest overlap. And then you think to yourself, these assumptions you have to work with here. You have to assume that these sequences are different. They're not the same sequence, and they do have some kind of overlap. And then you have to find that the longest overlap. And then you have to infer that that overlap is actually describing kind of what the sequence looks like at that position where those two fragments would be in the sequence. Right. So yeah, this is really what I'm talking about here. So you're looking at the suffix and the prefix of these two sequences here. Is the suffix and the prefix longer between these two sequences? Does it look like it's unlikely that those two chunks would, or the suffix and the prefix of these two sequences would be randomly the same? I mean, the longer the sequences are that are overlapping, then the more evidence you would have, I'd say, that would suggest that those two sequences do have an overlap. And so you have to kind of keep that in mind. One thing I will talk about, though, is that this is something that I find very exciting. One of my areas of research is to work with making graphs and networks about things. And if you're interested in hearing me talk about that, I can let you know. I can talk to you about my networks, which most people find a little bit overwhelming and slightly academic, and they don't want to hear it all the time. So I do understand. But what I'm trying to say, though, is that when you're working with overlaps, though, the way that you'd write your data is this way. So, for instance, we have these two sequences here. I'll just zoom in down here, where we have sequence 1 and we have sequence 2. And you can see that sequence 1 and sequence 2 is written in this blue table, if you will, TACCTTG. And by doing a whole bunch of alignments here. And by the way, when the sequences are small, then you can do your alignments in relatively no time at all. I mean, it's not a huge matrix that you have to build. Your n by m matrix isn't colossal, and so you're really just doing. I mean, you're creating all these smaller alignments, and so it's what I'm saying is it's within your computational reach to do that. Now, whether that takes some time, it probably does, but at least you can expect to get an answer. If you have if you're trying to align, for instance, a sequence which is 4 billion by 5 billion, a 4 billion sequence by another sequence which is 5 billion, forget it. Your computer is going to die before that alignment can be completed. But with these small fragments, these reads, it's within your computational grasp. So here we go. Here, what we see is sequence 1. We're looking at the sequence 1 line here. I'm looking at the blue box at entry number 1, TACCTTG. And I noticed that the sequence 2, which is right beneath it, these are arbitrarily listed, but I see that the sequence which is on line 2, which begins TTGAT, actually has 3 bases in common, as you can see right at the bottom here. There are the 3 bases are TTG. I'm looking at this part right here. And so the way that you write this is, I found it confusing when I first saw this, but you're saying on the line sequence 1, which is up here, I see this 2, and then in parentheses I see a 3, and then there's a comma, and then there's another entry with 3, parentheses 1. What kind of coordinates are those? Well, the first number is with sequence. That means that on the line where you're reading this number, it's saying the sequence on this line is related to the sequence on line 2. Okay? So that 2 right here is suggesting that you're looking at the other sequence here. So you're saying that this is a comparison between the sequence TACCTTG and the sequence TTGAT. Those are the two things, the two sequences. And the 3 in parentheses talks about the number of overlaps. How much stuff is in common with both of these sequences? And that would be something that would be found by your automated local alignment mechanism that you would have built into your algorithm. But what this is saying is that we have TTG, which is your at the end of your suffix at the end of your sequence, as we see right here, which matches the prefix TTG in sequence 2. And so when you see this, you're thinking, why would they write this in such a crazy way? But actually, how else would you write this? But I'll show you actually why you do this. So you're saying that sequence 2 and sequence 1 and sequence 2 have an overlap of 3. And the reason why this is important is because when you go to the next page, next slide here, you're building a graph out of the data. And so this is where it begins to get really blown back by the coolness, the excitement of the coolness of the whole approach here. And so each of these nodes here, 1, 2, 3, 4, 5, 6, 7, each of those nodes represents one of these strings, one of these fragments, one of the reads in our experiment. So for instance, node 1 is this TACCTTG. And what this is saying is, when you look at this number right here, 2, 3, 2 is an arrow or a connection between this sequence and the second sequence. So there's a connection between sequence 1 and sequence 2. And so what I'm talking about is this connection right, if I can zoom in here without my computer dying on me. But this 3 that you see right here, that 3, that is your connection. You're saying that sequence 1 and sequence 2 have a connection. The numbers between these connections, or the numbers on these edges, are the numbers of overlap that exist between these sequences. So for instance, 1 and 2, those sequences have an overlap of 3, and so 3 is your weight. So let's try another one here. So I'm looking at 1 and let's see what else we have here. Sequence 1 and 3 have an overlap of 1. I'm looking at the next available piece. I don't mean to make you feel dizzy if I keep zooming in like this. There's probably a trash can next to your desk, noise. Just kidding. But if it makes you feel dizzy for me to keep zooming in, then let me know. What I'm trying to say, though, is that you have another sequence for sequence 1. Sequence 3 also has some kind of connection, and that connection, or that overlap, rather, is just 1. And so I see, for instance, that the suffix and prefix of sequences 1 and 2 is T, and so that must be what that overlap is. And so let's see how that looks on the graph. And so I'm going to go to sequence 1 right here, and sequence 1, sequence 3 right here. So we have 1 over here, and we have sequence 3, and then the weight, which is this lonely number, right here, that 1 is the number of overlaps. And so what I'm trying to say is that you're building a whole network out of your results. So all these results from all the fragments, in this case we only have 7 reads that we're working with, but we accumulate all this data from those 7 reads here. So that's a bunch of local alignments that we ran, and from each local alignment we have this information which is then given to us so that we have an idea about what the overlaps are, which sequence has an overlap with those sequences, and what the overlaps are. But once we have that information, we build this map or this network, and then we can use basic network or graph theory type network traversal algorithms to figure out how do I get from network 1 to 2 to 3 to 4, or how can I get from one of the nodes to another node, such that I visit every single node I take every single path to get there. I mean, you work with different types of algorithms here to analyze this network, which is actually fairly commonplace in computer science, working with networks. But you're working with this network and analyzing this network, and the analysis of your network is the actual sequence that you put together from all these reads. So in this case, you probably would have some kind of network, which would be you have some kind of a greedy algorithm, as they call it, where you're trying to jump from node to node. You're writing a computer program which has what's called a greedy algorithm. It's an algorithm that wants more and more and more. This greedy algorithm starts at one of these nodes, and then it's trying to jump to the next node. But every time it does that, it's going to take the highest weight. So for instance, if our greedy algorithm started at node 1, it would go to node 2 because there's the weight of 3 on that edge between node 1 and 2 is higher than any other edge that exists. Node 2, for instance, I'm looking. I could go from node 2 back to node 1, but there's only 1 overlap there, and so that's not very much. But I see that if I go from 2 to 3, there's an overlap of 3, and so that tells me that this is actually much more exciting, so I'll take that because that's the biggest number. Remember, your algorithm always wants more. And the idea is that when you run this algorithm and you kind of stack together these sequences the way that using this greedy algorithm, then you can rebuild the original sequence. But one thing that I'll talk about, I'm looking for an example. Here we are. Now, remember before when I was saying that sometimes you get data that has this, you get genome sequences which the versions change? One of the reasons for that is because when they use a networking system like this, you'll find, for instance, look at node 6. Node 6, if you land on node 6, let's say you get to node 6 from node 5, where you take the greedy algorithm, takes the number 3, and so it goes from the edge of number 3 from 5 and it gets you to 6. Now you have to make a choice if you're at 6. Either you go to node 1, which has a weight of 1 to take you there, or you're at node 6, or you go from node 6 to node 2, which has another weight of 1. And so you have 2 matching weights. In other words, there is no greater number that you can choose. It's either 1 or it's 1. And so you have to arbitrarily decide. When I say arbitrarily decide, you have to kind of like put together some other, maybe some kind of tiebreaker knowledge, like, maybe there's statistics involved, maybe there's something else, but you need to tell the algorithm, hey, you're at node 6, where are you going to go? Are you going to go back to 1? Are you going to go to 2? Where would you find yourself? And so it actually does create quite a difficult problem here. In this case, it's not a very big problem because there are not very many reads, but if you have like, millions of reads, seemingly, then all of these nodes are going to be, it's going to be difficult to determine like how you can jump around exactly from one node to the other with the most greedy option. And so you'll find that there are multiple solutions, and then when you get a solution out of this map, out of using your align or your algorithmic approach, you will find that some of the solutions just aren't biologically feasible. That means you'll think to yourself, I know that this is a gene that this organism has in common with another, or there's a gene that this organism has which is in common with another organism. And when the data is like this, and I take these types of paths, I don't see that gene. The pieces are in the wrong order. And so you have to kind of liken this data that you're working with the data of another organism, and compare and contrast, and you find out which is the most likely of the genomes. And each time you do that, you're bumping up the version number in GeneBank because you're learning something new. And so you will notice that some of the genomes that are out there, the numbers seem to be climbing, especially some of the more complicated genomes of maybe some of the more difficult to isolate types of genes. For instance, if you're working with, I think if you're working with like mitochondria DNA, mitochondria actually has its own DNA. That's the energy producer of your cell. It was thought that mitochondria was its own cell that took up residency in the regular cell, in the eukaryote cell. But mitochondria does have its own DNA, and when you're trying to sequence mitochondrial DNA, it's very difficult to do logistically because it's very delicate, and so it's hard to do that. So when you're making these maps, you might find that there are so many errors introduced that your maps are. You may have to make hundreds of different maps and then do the analysis over all those hundreds of maps to figure out which map was it that had the fewest logistical errors. Yeah. So anyway, that's a side point here, but mitochondria is fascinating. There are there's really two basic techniques that we've just discussed here, where you're looking at kind of like this overlapping system where you're just looking at the reads, you're cutting them, that have been cut out of a regular cloned piece of DNA, and then you're figuring out what's called the consensus, and that's at the bottom here. And the consensus is really a word that describes, when you put all these pieces together, where are what's the final sequence going to look like? And so this sequence that exists on the bottom of this thing here, this CGATTCTAAGT, that's the sequence which is built out of the combined overlapping fragments. And so that's what they call the consensus. And so that's pretty cool. Now, the other way of doing things, that's if these, by the way, these reads, the only big difference here is that these reads that we're looking at here are naturally cut apart. You use restriction enzymes, which are basically tiny little microscopic, or they're like little scissors, I guess, that cut the DNA at specific regions, specific places. And so that's how the DNA is shredded like this. But you can use something else called the K-mer system, and K-mer, the K is, it's a variable for a number, and so you're saying that I'm going to cut up the pieces of my sequence into all words of a particular length. I want all the same length. I want to work with, in this case, I'm going to work with all fragments of length 3. And so when you're working with the K-mer system, then that means that you can actually control the size of these reads. On the other side, on the left-hand side, you don't control the size. Nature controls them, if you will. Wherever they get cut, that's the size you get. And so the sizes on the left can range from 1 to thousands of bases long. Whereas on the right-hand side, using this K-mer, where K could be 3 mer or 4 mer or 5-mer. The K actually means a number, and so in this case, this is a 3-mer. But the K-mer system is that I'm going to make sure that my system or my computer is going to be able to deal with this data, because I know that every single read that's going through that I'm processing is length 3. So there's no cases where my computer just gives up because it's working with a I don't know, 4,000, 5,000, 6,000, whatever it is, length chunk of DNA, which is the read. Maybe my system doesn't work that way. Maybe it doesn't have the, I don't have the computational power to run that kind of analysis. Whereas if I say that everything is 3, then I do. And so basically, in that case, you're making these k-mers, and then you're building a graph, just as we saw with this data here, and you're building a graph like this. And then what you want to do is you want to do what's called walks on those graphs. And so, it's like you're basically starting at one of these nodes, and you're trying to walk to all the other nodes. And by the way, each node represents a 3-mer, or a word of 3 bases. And you're trying to walk around and hit every single node in your map. So in other words, you're making this into a game where you're trying to start at one of these nodes here, and whether it's the greedy algorithm or there's something else, but you're trying to visit every single node on this map, and they call that walking maps. You're walking the graph. So actually, next time if you're on Tinder or something like that, and somebody asks you, like, what do you like? You say, I don't like going for walks on the beach. I like going for walks around my maps. It just sounds better. No, it doesn't. Okay, so I'm going to move on now. So that's basically the end of this. I'm going to that's the end of our slides. So I'd like to now do something, which is I'm going to try something a little bit different here. In your class docs, you would have found this activity. This is activity 9. God, we have a lot of activities in this class. It's very exciting. I'm going to try something that's slightly different. I know that this looks like a lab, and I dressed it up to look like a lab too, but this is a discovery piece for you. As we discuss technology in this class, we usually find some kind of a website that demonstrates that technology. And so in this work here, there is actually, in sequencing, there is a technology which is available online, which is called this EG assembler. An EG assembler allows you to dump in the reads from like all these these pieces here, read 1, 2, and 3, and then it will actually give you the sequence. It's magic how it works. It truly is magic. You just pour in a whole bunch of sequences, and then those sequences are then put together like a jigsaw puzzle using the EG assembler's algorithm. I mean, it's just incredible how you see this working. So what I'm asking you to do is, I don't want to say too much about this thing here, because I want you to I want 2 things here. I want you to discover this tool for yourself, but also, though, one of my interests is that as we're getting closer and closer to the end of this class, you're going to start. We'll talk about our projects very, very soon. You're going to have to go out and find tools to work with your projects. I mean, you'll find tools online. You'll find maybe different packages of programming. You'll be working on some small research project of your own. And we will discuss all that later, but you're going to be teaching yourself how to use tools. And so I'm going to try something different this time, where I want you to discover this tool for yourself and learn how it works, and then to write everything up. So I ask you to run this, there's a survey here, and it's called, I call them surveys, but actually it's just, it's some questions here. And these questions are going to be, you'll find as you run through these steps here to use this tool, the questions in blue, or the questions that you have to respond to, are embedded in the tasks themselves. Now, if you go to the form, actually, can I do that here without crashing this? Yes. So if you go to the form here, actually, this should be just, actually, this is actually 9 here, not 8, but I'll change that. If you go to the form, you'll find that these questions that you're supposed to respond to are actually these pieces that you're supposed to, or that you find in the steps. So I'm going to end this here. We can get out of class this a little bit earlier today, but go ahead and run through these steps here. You will find the data that you'll use for this in a directory here called data reads-fasta.txt. Typically, what I would have done with this file is just call this reads.fasta, and fasta would have been my extension. But I recognize, though, that it's nice to be able to click on a file and have some established editor in your operating system load this file and open it up as a text file. And so I've just saved it as txt. But typically, though, when you download these files from online websites, it's just .fast. And so that's why it looks a little bit different. Yeah, so anyway, go ahead and just go through these things here. As you know, I'm available on Slack. You can always send me a message. You can speak to each other. But I think have some fun with this here and to see what this tool is all about. You'll find that, for instance, this EG assembler is just magic. It puts together like these giant jigsaw puzzles in like moments. It will take some time, though. I'll just say one more thing here. When you're running, in fact, I'll just see whether this is, it seems to be loading pretty quickly. But one thing I will say, though, is that I did this lab actually last night. And when I was working on this last night, it seemed like it was much more when I was working on it yesterday around, I want to think, like in the morning sometime? Let's see. So anyway, but what I'm thinking is that this is a website which is located in Japan, and so maybe, for instance, the servers are going to be busy at different times. But there are, I mean, you'll notice that if you use these tools enough, you'll realize that there are times of the day when, I mean, forget using them because it's everybody else is using them. So you just know that, oh yeah, it's 3 in the afternoon. Now it's time for my blast search on NCBI. You know, it's that kind of thing. Or maybe it's 1 in the morning. It's time for my EG assembler analysis. And so keep that in mind. But I think that one thing nice thing about this website is that you can add your email. And they're not going to ask, or they're not going to do anything weird with your email. But you can add your email here and a project name, and then they can actually send you they'll send you a link to the results, which is actually quite nice. So you don't have to actually stand there and wait for this thing to finish, but rather they'll send you a mail and give everything. Now, when I did this, I don't think that there's any trouble with putting my email out there, but I do understand if you don't want to do that, that's quite all right. I mean, they're not going to give your email address to your, you know, your arch nemesis or something. I don't think they're just using it just for this. But anyway, if you have any questions, I can be reached on Slack or I have office hours and anything else. This is actually due. This activity is due on the 16th, so that's Monday.